what this session is about is, well, disrespectfully, the other approaches, but maybe you can think of it more as the outside of the box approaches. We know it's really difficult. We can restore dystrophin, but there's lots of other things ongoing also in the muscle. The muscle has damage, the regeneration is impaired. So we need to think outside of the box to treat this, and we have 3 presentations on out-of-the-box approaches to try and improve the muscle quality. And the first presenter is Linda Marban from Capricor Therapeutics, and she will present on their out-of-the-box approach first.
Marban will be surprised
I was going to say, you don't look like a Linda.
Because if you know Linda Marban, you know that I'm not Linda Marban. But my name is Dan Paulson, and I work for Linda Marban. I am the Vice President of Development for Capricor Therapeutics, and it's my pleasure today to come to you to talk about one of the other, or outside-of-the-box therapeutics in the indication space for Duchenne. I've really enjoyed this meeting so far, and it's really a pleasure to be here and talk to you today about CAP 10002. Let me find my slide advancer. These are my forward-looking statements. The most important one is that CAP 10002 is an investigational product. We're using it in clinical studies right now, and it's not approved for use in Duchenne's. So CAP 10002, what is it? It's one of the other therapies, but essentially, CAP 10002 is an allogenic, cardio-derived cell, and it comes from donated hearts. So these are healthy donated hearts that essentially aren't transplanted for technical reasons. And so it's not gene therapy. This is not a stem cell, but this is a specific cell line within a heart tissue that actually works when infused into the body. It's sort of an allogenic transplant. It works by communicating with other cells. And so it's a pretty cool technology. It communicates via exosomes. And 20 years ago, we thought exosomes were basically the way that cells got rid of trash. But we know now that they're actually a fairly effective way that cells communicate with other cells. And so what the exosome is, if you think of like a big hot air balloon, an exosome is essentially a golf ball that comes out of that hot air balloon and will move and migrate and then basically will fuse with another cell. And it transfers contents from one cell to the other. And for example, those contents may be microRNA. And in doing so, it actually has its mechanism of action. And the target cells that it tends to fuse with, skin cells, connective tissue cells, white blood cells, but importantly, muscle cells, skeletal muscle cells, cardiac cells. And so it has an anti-inflammatory, anti-fibrotic, regenerative effect on the cells that it fuses with. And so we've looked at CAP 10002 in many, many patients, but the majority of them with Duchenne's. And I have 4 messages today that I want to deliver to you. The first is, what's CAP 10002? The second message is, I want to show you some of the data from our phase 2 program, HOPE-2. The third message for families and patients and investigators, I want to tell you about our phase 3 program now. And the fourth message is, stay tuned because we have a late breaker this afternoon. So what's our target patient population? I sat through 3 hours of discussions last night about the science, and there is sort of a target patient population that right now is sort of the orphan disease within Duchenne's, and that's our older patient population, the non-ambulatory patient population. And that's exactly our target patient that we're focusing the HOPE development program on. We're looking at young men, 10 and older, who are non-ambulatory. And our endpoints that we're looking at are performance of upper limb and cardiac endpoints. But really, our patient population is a patient population that many of the other sponsors or programs that you're hearing from today aren't focused on yet. But we're exclusively focused right now on 10 or older, non-ambulatory. So what have we done so far? Well, we've done several programs within that we've completed. Hope Duchenne, which was the initial program, sort of our phase 1/phase 2, looking at CAP 10002 in young men with Duchenne, and the results looked very intriguing. We were dosing a little differently. We were delivering a little differently, but we learned from Hope Duchenne, and we adjusted the dose, we adjusted how we delivered, and we moved the program into Hope 2. We've completed Hope 2, and I'll share with you the data today. I mean, I'm excited about it. Hopefully you'll be excited about it. And now we've also continued, we've rolled the patients from Hope 2 over into OLE, open label extension. We're going to show you this afternoon, Dr. Marban will show you this afternoon, the 12-month data, but we have these young men now for 24 months almost, and we're going to move to 36 months. And then we've just opened Hope 3, our phase 3 trial. So the results of Hope 2, and why we're probably off your radar screen, the results of Hope 2 were just published 3 months ago in The Lancet. Just published 3 months ago in The Lancet. So you can pick this up, you'll see the reference below. It's a very interesting article. Again, a very independent, external review of the work. And I'm going to share with you some of the data now. So I'm going to move right to our endpoints. So Hope 2 was basically a double-blinded, randomized study. The endpoints were the PUL 1.2. Analysis was done at 12 months. And we also looked at cardiac MRI for EF, analysis at 12 months. We enrolled 20 patients in Hope 2, and we followed them out for the entire year to the primary analysis at 12 months. And you'll see here, the PUL was our primary endpoint, performance of upper limb, and you see very quickly, a separation between active treatment and placebo. And you see that that separation over the entire year continued. In fact, we received statistical significance at 6 months. And the p-value at the end of the 12-month period was 0.1 with 20 patients. This equated to about a 2.6 change in the PUL score. And it was a 71% decrease in the progression of the disease. The cardiac endpoint is to the right. We looked at cardiac MRIs, and the EF was really our primary endpoint with the cardiac MRI. On average, these boys started at 54% when they came in. Numerically, at the end of 1 year, numerically, these boys had improved function. Their EF actually increased by 0.1%, whereas the boys on placebo decreased by about 3.8%. That p-value, as you see, was 0.002. I'm not a statistician, but I think what that means is this didn't happen by chance. So in terms of the safety summary, this is a transplant, an allogeneic transplant. We did see some infusion reactions early in the program. The program was put on a voluntary hold. Recommendations were made for a prophylactic steroid and antihistamine infusion treatment the day prior. And after that, there was really very little of that infusion reaction that was noted. And now I'll remind you that we've given 90 extra doses basically in the OLE program without issues. So that leads me to the phase 3 program. Phase 2 data looks good. At least we were pleased. And so now we've designed and worked with the agency on our phase 3 program, which is called Hope 3. Hope 3 actually is open for business. We have several sites now that are open for enrollment across the US. Very pleased to visit with several of the families and patients in here at our booth. And very excited to visit with a few of our investigators from Hope 3. But let me tell you a little bit about Hope 3, and I hope I don't get the 15 minutes walk up. But Hope 3 is, again, a randomized, double-blinded study looking at CAP 10002 compared to placebo in a Duchenne population. There's a screening phase in which the boys will be evaluated for eligibility. If they move through the screening phase and they're eligible for the study, there is a day 1 dosing. And then in HOPE 3, the dosing is just like HOPE 2, every 3 months. And we'll do functional evaluations every 3 months, including the PUL 2.0, which is the updated performance of upper limb. And the primary analysis will be done at 12 months. We'll have cardiac MRIs at 12 months. We'll have the performance of upper limb 2.0 at 12 months. And this will be the primary endpoint for the phase 3 program, HOPE 3. We will roll these boys directly over into an open label extension. So after the primary analysis at 12 months, every boy, regardless of whether they received active treatment or placebo, will get open label active treatment, CAP 10002, for the next 12 months. And just a reminder that in HOPE 2, you'll see the open label data this afternoon for the first 12 months. How many patients will we look for? About 70. And we're doing that at 25 sites across the US. So what's the eligibility criteria? Again, the focus of this program is boys over 10 years old with a confirmed diagnosis of Duchenne and essentially a non-ambulatory or in that transitional phase with PUL scores or the classification of 2 to 6. HOPE 2 had a classification of 2 to 5. We've expanded now up to classification of 6 for our target patient population. And we're focused on Duchenne certified, Duchenne care centers because we want to assure that the standard of care for these boys is the same and very consistent. We're looking for patients who are on stable doses of steroids. And essentially, this is a long list of eligibility, but what we're looking for is stable patients with disease states that really will not confound our outcomes or our endpoints in the study. So for example, we're looking for boys with MRIs with EFs greater than 35%. BMIs greater than 45% could interfere with some of our measurements, so that's an exclusion. But essentially, we're looking for young men with Duchenne's, 10 and older, non-ambulatory or in the transition phase, that are on a fairly stable, plateaued area of their clinical course. So in essence of what my messages are today, one is I wanted you to learn a little bit about what CAP 10002 is, how it works, why we're excited about it being studied in Duchenne. I wanted to share with you some of the, what I consider exciting phase 2 data. I wanted to tell you a little bit about HOPE 3. And again, set the stage. At 5 o'clock today, we'll have a late breaker. We'll share with you the results of HOPE 2 OLE 12 months, and Dr. Marban will share that with you this afternoon. So thank you for your time today.
Thank you. If you can please take a seat on one of the chairs so we can have Q&As afterwards. And well, you're leaving us with a cliffhanger, but we have something to look forward to later this afternoon. So the second out-of-the-box approach is from Astellas. The presentation will be given by Tran, and I think, if I remember correctly, it is something to do with mitochondria, but if not, I'll stand corrected.
Thank you so much. Good morning, everybody. Thank you so much for being here on a Saturday. My name is Tran, and I'm a board-certified pediatrician. I've been working in the rare disease space, rare pediatric disease space for quite some time, but I am new to Duchenne, so thank you for welcoming me. And I am a senior medical director at Astellas Pharma in the global development department. And so Astellas is a medium-sized biotech company, and the headquarters, US, is located in Chicago, Illinois. And we also have a branch in the main headquarters in Tokyo, Japan. So I'm one of those remote workers. So I work from the Silicon Valley in California. So kind of reverse of what most people do. Most people will try to go somewhere rural and less expensive, but I'm on the other side. So this is my first in-person event since early 2020. So it's great being here today. So for disclosures, I do work for Astellas. I am employed by them. ASP0367 is an investigational drug and not available for use outside of investigation. The safety of efficacy of the molecule has not been established, and there's no guarantee that the molecule will receive regulatory approval or become commercially available for uses being investigated. It is also not authorized for sale in any jurisdiction. So now I want to give an overview of ASP0367. We also refer to it as Bossi-Delpar in some locations, and its role in affecting the mitochondria. So ASP0367 may act inside muscle cells to increase the mitochondrial's ability to make energy and improve muscle health and function. Mitochondria is an organelle, or a small organ of a cell, and it plays a vital role in production of ATP, which is what you need to make energy, and it's critical for muscle contraction. So why is mitochondria important? Well, it's present in every organ in your body has cells, and every one of those cells have mitochondria. So in fact, every cell has approximately 100,000 to 600,000 mitochondria. So in the normal muscle, as you can see in the upper photo, a network of functional mitochondria ensures the proper energy production to meet the requirements of the active muscle. So in Duchenne, the loss of dystrophin impairs mitochondrial function because dystrophin provides the muscle fibers resistance to mechanical stress. So if dystrophin is deficient or does not function well, the muscle fibers are more susceptible to damage, and the muscle fibers become structurally unstable and more porous to the environment. So this leads to excessive calcium influx, which can lead to calcium overload, which contributes to inflammation, fibrosis, and you'll have muscle dysfunction and eventually muscle degeneration. So ASP0367 is taken by mouth, and it can be given in combination with approved therapies such as exon skipping therapies and corticosteroids. So one important thing to understand is that this molecule was granted orphan drug designation by the FDA in March of 2018. So what that means is that after review of our non-clinical and clinical data, the FDA identified ASP0367 as a promising therapeutic for development in this rare disease. So I won't be presenting our non-clinical data or our clinical phase 1 healthy volunteer data today, but if you have any questions, our investigators do have the investigational brochure. And if you are a physician that is considering referring one of your patients, I'm very happy to have a one-on-one call and have myself or other team members walk you through that data. So we have, in terms of clinical studies, so we have completed a phase 1 healthy volunteer study, and that was reviewed again by the FDA and contributed to achieving the orphan drug designation. And then we also have ongoing phase 1b studies in patients with low systemic oxygenation, often diagnosed with chronic fatigue syndrome. Additionally, with this molecule, we are also in a seamless phase 2 to phase 3 study in primary mitochondrial myopathy. Okay, so now I'll briefly go over how ASP0367 may improve muscle energetics. So when the molecule is taken, the body sends a signal in a pathway called PPAR delta. It's also, that's short for peroxisome proliferator activated receptor delta. So that is turned on, and it's a group of nuclear receptor proteins that function as transcription factors, and they transcribe and regulate mitochondria by turning on different genes in the cell. And so in our phase 1 healthy volunteer study, the genes that were found, they were found to be upregulated, meaning that they had increased gene expression. And those genes were fatty oxidation genes such as CPT1, carnitine, palmitoyltransferase 1, and PDK4, pyruvate kinase dehydrogenase 4. So the signal has been turned on for the mitochondria to use more fatty acids to make more energy and potentially improve muscle health and function. So because of this unique mechanism of action, which is independent from others that you may have been hearing about, it can be used with exon skipping therapies and or corticosteroids. So this makes our clinical trial quite inclusive of other therapies. So as mentioned before, we do have a phase 1b randomized, double-blind, placebo-controlled trial. So the first 12 weeks of the trial are randomized double-blind, and then the second 12 weeks is actually open label. And we'll go through the study design in later slides. So the primary purpose is to evaluate the safety and tolerability and early efficacy of the drug. So in terms of the endpoints, the primary endpoint is safety. The secondary endpoint is PK/PD, so really studying what the drug does to the body and what the body does to the drug. And then we also have secondary efficacy endpoints as well, where patients are, we are assessing the number of times a patient does this assisted cycling test. So they'll cycle for 6 minutes and see how many times they can do that. And then also the PUL 2.0. 2-minute walk test for ambulatory boys, and a few patient-related outcome measures. So we have 9 sites open and now recruiting in the United States. The cities are listed there: Los Angeles, Sacramento. It's actually not Kansas City, but Kansas, where the University of Kansas is. Worcester, Massachusetts, Baltimore, Minneapolis, Philadelphia, Norfolk, Virginia, and Richmond, Virginia. If you have questions about which sites, if there's a site near you, we do have a QR code and a clinical trial website, and the information was provided in your registration red bags. So here's the geographical map of our sites, and we do realize that there are disparities in terms of the locations, and often it is due to the fact that they are all children's hospital, and we understand that the resources are limited at the universities. But we really want to give a special thanks to our sites, so UC Davis, CHLA, University of Minnesota, University of Kansas, CHOP, Carnegie Krieger, who has a very strong presence here at PPMD, and thank you. They are our top enroller, so congratulations to them. The Children's Hospital at the King's Daughter and Children's Hospital of Richmond at VCU. And then, because the locations are scattered, if you do not live close to a clinical trial sites, the majority of our sites are accepting referrals, and Astellas does reimburse for travel and meals for 2 caregivers and the patient as well. Okay, so who can participate? So we are targeting a total sample size of 18 patients, boys ages 8 through 16 years old, and you could be ambulatory or non-ambulatory with some caveats. So for ambulatory boys, we are asking that they have some trouble walking. So the 10-meter walk test, it needs to be completed longer than 6 seconds. And then for non-ambulatory boys, we're looking for the PUL 2.0 score of 4, 5, or 6, so meaning ability to independently raise your arms above the shoulders. Patients will need to be taking a stable dose of corticosteroids for 6 months, or not taking steroids is fine as well. And then key exclusion criteria are any acute illnesses, and the PI will assess whether or not the patient may not be healthy enough, either physically or mentally or behaviorally. So in terms of cardiac health, we are currently asking for essentially normal heart function with ejection fractions no less than 55%. We are excluding patients with a long QT interval. And then the troponin levels have actually been edited. So we will be trending them in the clinical trial, but we will not be using them as inclusion-exclusion criteria unless the principal investigator feels that it is abnormal due to a pathological cause. The use of coenzyme Q within 4 weeks is prohibited, and participants need to have adequate renal function. Lastly, again, patients who are receiving an investigational drug that is not FDA approved will not be eligible for this trial. Okay, so here's a little bit about the study schedule. So we have a 4-week screening period and then 12 weeks of a double-blinded portion. So it's randomized 2 to 1. So we anticipate 12 patients to receive the active compound and then 6 patients to receive a placebo. And then after 12 weeks, everybody moves on to the active drug, and we have a low dose and high dose for both portions of the study. And then there is a 4-week follow-up period. So as you can see, it's a relatively short trial. And then there is an option at certain study sites to do home visits as well. So it could decrease the burden of travel. Okay, lastly, we do have a pre-screener tour, as I mentioned before. You can scan the QR code that's available in your conference bags. And through there, there are different questions that you can see if you or your son may potentially qualify. And just a little bit information more. We have a website. We're listed on clinicaltrials.gov. We have a QR code. We really want to thank all of our patient advocacy partners, including PPMD. Additionally, I really want to thank our patient ambassador, Mrs. Dana Edwards. I find her to be extremely inspirational. She is the inventor of the perfect lift, and she's been working with us to ensure that our clinical trial design fits your needs, and that we are being as inclusive as possible. So thank you so much, Dana. And I've yet to meet a single person who does not know who Dana is. So if you have any questions about the trial, please feel free to also reach out to her as well. Thank you very much.
Thank you very much, and also for both sticking to time. So the last talk in this session is from Joanne Donovan from Edgewise, and that is an approach to make muscle fibers more resilient to damage. So you also have 15 minutes, and then we have time for questions.
Thank you so much. It is truly wonderful to be back here live with this community, and I'm thrilled to be here on behalf of Edgewise. So Edgewise 5506 is a novel approach to protecting muscle in Duchenne muscular dystrophy. And first, I want to just tell you my disclosures, a admittedly unreadable forward-looking statement, but importantly, EDG 5506 is an investigational agent, and it's not approved in any territory. Now, I'm just going to give you an overview of our thinking on this. And I think that the biggest thing is, as you well know, when boys are infants and toddlers, they actually aren't diagnosed right away. And they're not diagnosed because their muscles are functioning. They're able to contract their muscles, and it takes a few years. And that few years is because the damage is accumulating. So what we know and what we're trying to use to develop a good therapy is that some muscle fibers are more susceptible to injury because of the lack of functional dystrophin. So what we've done is make an investigational agent, EDG 5506, that's designed to protect the most susceptible fibers from damage. What we've seen in diseased animal models is that EDG 5506 protects susceptible muscle fibers and prevents longer-term development of damage. So we've been able to take it into the clinic, and I'll show you some data from the clinic as well that supports this approach. Now, what does dystrophin do? In every talk you hear about dystrophin, right? But what does it really do? People talk about it as being a shock absorber. It's not a contractile protein. It doesn't contract the muscle. What it does is protect the muscle from stress during contraction. And it's shown here, and there are these little bridges, these yellow bridges that are pointed to with dystrophin. And what those do is that it connects the contractile machinery, the proteins that actually do the contracting when a muscle contracts. It connects it to the outside of the cell, the matrix of proteins outside the cell, and then to the next cell. And that cell is then connected to the next cell. So what that does is it spreads the stress of contracting a single fiber. It spreads it over the whole muscle. Remember, muscle fibers don't all fire at once. It's a small subset of them that fire to contract your muscle. So what you don't want is those individual ones not having the support of the rest of the tissue. So mechanical stress, we know, damages dystrophic muscle. In fact, you know and you hear from your doctors that contraction when lengthening the muscle is particularly damaged, so you need to watch out for that. Eccentric contraction, it's called. And when dystrophin is lacking, the mechanical stress that occurs during contraction damages the muscle. What's seen here is that they're actually under the microscope, you can see that the fibers become damaged, they become jumbled. The membrane becomes leaky and allows biomarkers to go out into circulation that we see clinically in diagnosing the disease. So basically, this damage, when it continues, leads to fibrosis, it leads to functional decline. Now, what we're hoping to do with EDG 5506 is to prevent the mechanical stress from causing that damage. And what we're doing is modulating the contractile fibers. And then even in the absence of dystrophin, then that protects the muscle fibers. And what we hope is that that protects the muscle over the long term and allows maintenance of function. Now, the next slide shows what happens in a mouse muscle. So I'm not going to show you a lot of detail on animal models, but what this is basically a finger muscle of a mouse. And it's contracting on a regular basis. The one on the left is from an MDX mouse, and you can see that what starts to happen as the muscle is contracting is there tends to be these little globs of protein, basically. The calcium comes in and causes cell death there. But it causes visible damage that you can see in that muscle fiber, in that muscle, actually, composed of fibers. When we pre-incubate that muscle with EDG 5506, you can see on the right, the muscle's still contracting, but it's not getting that damage with mechanical stress. So that's what we're trying to do here. And we've seen in longer-term experiments in MDX mice, in golden retrievers, decrease in the biomarkers and decreases in fibrosis, improvements in strength over the long term. So one of the indexes that we are able to use in clinical trials is looking at biomarkers. You know that CK was one of the first things when your son was diagnosed. And when the muscles are injured, the membrane is damaged, and it releases biomarkers like CK, things that are normally inside the cell that are leaky and are now in the bloodstream. Creatinine kinase, myoglobin, fast troponin, which is very specific for the kind of muscle fibers that are damaged most easily, and other proteins that we can look at. So we did a phase 1 study in unaffected adults as well as in adults with Becker muscular dystrophy. And what this tells us kind of answers some very important questions. What about the side effects? And what we saw over 2 weeks of dosing is that the molecule was safe, was well tolerated. Now that's only 2 weeks. We need to look longer, but it's a good start. How is it given? How is it absorbed? And we found out that it can be given as a tablet. It's well absorbed whether you take it with meals or without meals. And it can be taken just once a day because it lasts a long time in the bloodstream. Importantly, does it get to the muscle? Because we've heard a lot earlier this morning about making sure that specific drugs get to the muscle. That's the hard part sometimes in drug development. And what we found with muscle biopsies is that EDG-5506 is highly concentrated in the muscle compared to the bloodstream, 100 times more concentrated in the muscle, which tells us that it's getting to where it needs to be to protect the muscle. And we saw that both in the unaffected adults as well as the patients with Becker. We saw a much higher concentration in the patients with Becker. Now, what happened to the biomarkers? And what this shows is in the first column for creatine kinase, CK, and for fast skeletal muscle troponin, specific to those most susceptible fibers. In unaffected adults, it's low. At baseline, before they were treated with anything, with the Becker patients had high levels for both. Now, when these guys, and we need to give a shout out to them, they were in a phase 1 unit for 2.5 weeks, which means they were not great accommodations, not great food probably, and they were confined. They weren't able to move around very much, and we really appreciate how their sacrifice in doing this. So what happens when you don't exercise as much is your CK goes down a bit. And that's actually what we saw in the placebo in the third column. Both the CK went down a little bit and the fast skeletal muscle troponin. But the patients who were on EDG-5506, after just 2 weeks, we saw a pretty remarkable decrease in the CK and in the fast skeletal muscle troponin, down to levels that were pretty close to normal. So we were very excited to see this. And what we did was we had an open label extension study so that the 7 adults with Becker were able to, they went off the drug for several months, they came back, they started dosing again in January. So now we have information, not just with 2 weeks of information, but with several months of information. And earlier this week, we shared that after 2 months, we see, again, the CK going down, the fast troponin going down, despite the fact they're out and about and doing everything they're supposed to be, not just in a unit. So we're very encouraged with that, with those results. And again, it seems to be safe and well tolerated over this period of time in these patients. Now, why have I told you all about Becker? We are here to hear about Duchenne. I mean, this is a spectrum. You've heard that several times during the meeting, that it goes from complete absence of dystrophin to partial absence of dystrophin or functional dystrophin in Becker. So this is the same disease. It's a spectrum. So if we can protect the muscle fibers in Becker, then we also believe that that will be the case in Duchenne. So we have started in Becker. We're moving into Duchenne later in the year. Now, overall, our goal is to develop an oral medicine that will preserve and improve function in Becker and Duchenne. If we can prevent the damage, then we can allow the muscle to regenerate. We can prevent it from functionally declining further. And this has the potential to be used alone or also in combination with other therapies. So all the exon skipping therapies, and potentially other therapies like microdystrophin that replace some form of dystrophin to the cells. So alone or in combination. And really to target the initiating event, the mechanical stress-induced damage to these cells. So I told you about the ongoing open label study in these patients that were started with Becker. We've also started a natural history study in Becker. There's much less information. And we have funded the GRASP network to do a natural history study in combination with imaging DMD. That's ongoing, open for enrollment. We've started a phase 2 ambulatory study in ambulatory adults and adolescents with Becker that is soon, you'll hear more about that, soon to be enrolling, and starting an initial study in ambulatory boys with Duchenne to start in the second half of this year. A little bit more about that study. We are planning on enrolling boys from 4 to 9. They will need, of course, to have a confirmed mutation in the dystrophin gene with the characteristic phenotype, ambulatory, and on a stable dose of corticosteroids. They can also be on a stable dose of exon-skipping therapies that are approved. This will be a 3-month placebo-controlled study with an open-label follow-up period of 9 months and hopefully extended after that. And we can take advantage of the fact that we can see really rapid changes in biomarkers to understand what's happening in these boys and to select a good dose for the phase 3 study. So we can really take advantage of that and minimize the placebo period in this initial study. We'll be looking at safety, biomarkers, pharmacokinetics, how the drug is absorbed, and we'll also be looking at functional assessments. But the major endpoint of this study is to look at biomarkers in order to be able to design a bigger registration study. So I'd like to thank you all for listening. Special shout out to the adults with Becker who participated in the study and are continuing to participate, as well as the advocacy organizations that have really supported how to do clinical trials and made us do better clinical trials over the years. So thank you.
Thank you very much. And also thank you everyone for sticking to time. You'd almost think that exon skipper people have a tendency to run over their lot of time. And the people that think outside of the box will stick to time. So thank you very much, because now we do have 15 minutes for questions. I'm not sure whether there's questions online or whether there's questions from the audience already, because I see...
No questions online
questions
yet, but I do have a question. Just to confirm, a number of you mentioned that you'll be allowing for those who are on approved exon skipping drugs to be part of your studies. Can you all confirm that? Yes. Will you analyze that subset differently than those not on when it comes to your final analysis?
We will not for phase 1B. No.
And we will certainly look at those patients separately. Basically, those patients are being turned into a form of Becker. So again, it goes to the continuum of why we're looking at those patients as well in the study.
And we will also. It's important to realize that they'll be on stable doses to come into the CAP 10002 program, but we'll definitely look at them as a subset.
Thank you.
There's a question over there.
So my question is for Dr. Donovan. First of all, amazing graphic. It was really cool, and I especially like the fact that you're concentrating on biomarkers instead of for assessment. My question is more around standard of care. I believe most Becker patients are not on corticosteroids. So how do you see do you see any mechanism of action overlap with steroids and your drug? And have you sort of looked at that?
So my understanding of your question is Becker patients are not typically on steroids, so how do we look at that with or without steroids? And so we will have the opportunity to see how the drug works in Becker patients. Teenage boys will also be included in the phase 2 study versus patients that are on steroids in the Duchenne study, because based on the mechanism of action, it should work with both of those.
Okay. Thank you.
Okay, and I have a question as well for every one of you. So, will there be biopsies in the trials in the phase 1, phase 2, phase 3? Will they be there or not? And maybe just starting on the left.
Okay, so for the Astellas trial, no therapies, no infusion. I'm sorry, no biopsies, no infusions, nothing invasive. We do have some PK/PD monitoring to be done. So those are blood draws, and when they actually have to stay a little bit longer, the sites do have an option to put an IV in to keep the fluids running and do those blood draws at those certain time points.
No biopsies in the studies.
No biopsies.
Perfect. Thanks for clarifying that. We have another question.
Yes, hi. I have two boys, ages 9 and 13. They were both dosed with gene therapy in early 2020. So I'm just wondering if they would qualify.
So we do not exclude patients with gene therapy. I think we would need to review on an individual basis.
It's such a tough question because gene therapy, as we've heard, is certainly adding back dystrophin, but this disease needs a combination. So it's something that we've talked about and how to include those boys in our studies going forward. In this phase 2 study, that would be an investigational agent, so they won't be included, but it's something that we're really putting a lot of thought into what to do next.
And from our standpoint, we're 10 and older. So the older child would be considered for HOPE 3, but a very similar answer to Edgewise. In terms of the gene therapy, we would have to really look at when the child was enrolled, whether they're still being followed, in terms of the therapy. We are excluding investigational products, and this is very typical in clinical research, not to be in 2 trials at one time. So simply answer your question, 10 and older, and then it would be much like the Edgewise discussion. We would look at it in terms of exactly what the gene therapy program was.
Thank you.
And we have a question online.
Joanne, do you have any information in regards to cardioprotective effect and or muscle impact in the heart or diaphragm?
That's a great question. And what we have seen in the preclinical studies is that although the molecule doesn't directly interact with cardiac myosin, so it doesn't do anything to cardiac contraction, we see in the mouse, the long-term, most severe mouse model, that there is a decrease in fibrosis in the mouse model. So it is something that we are following with echoes in these studies and will be following in the clinical studies.
And I had a question just intriguingly because I heard that your cells are isolated from hearts. And then, so how many cells can you isolate from heart, and is that a supply that, I assume it's a limited supply, so are there any concerns there, or do you say, well, that is not a concern? Just a logistic question from my end.
For manufacturing? It is a very interesting question in terms of donated hearts and then the manufacturing of CAP-10002. And I can tell you that initially with the company, as we're ramping up to assure clinical trial supply, we are able to really meet the clinical trial supply. And I know that the manufacturing group is really looking at ramping up not only, for example, Cedars-Sinai facilities, but also internal facilities to make sure that as CAP-10002 moves towards approval, that we'll have plenty of supply. It's a very complicated process, and actually Linda might be the best one to, since it's manufacturing.
Maybe it's a bit scientific, but I was just intrigued because I didn't know that they were isolated from heart. Linda.
So this is one of the most key questions, right? Will supply be able to meet demand? So CAP-10002 is a product that is made from isolated hearts. They're hearts that are transplant qualified, but can't be used for technical reasons. To give you perspective, we started in the field looking at making CAP-10002 for adult heart disease, looking at making hundreds of thousands of doses per year, and the scale up is absolutely possible. It's an isolated cell in the heart that we have patented methods to get out and then expand, and then the expansion is the scale up that Dan's talking about. So we have internal manufacturing capabilities. We've been manufacturing these cells for clinical use for about a good 8 years now. And then we're also building a new facility in our San Diego labs that will be able to meet initial commercial demand, as well as exploring other opportunities as the demand exceeds our internal capacities.
So one heart it's not one heart, one patient, it's one heart, multiple.
Yeah, we're estimating several thousand doses per heart, and that will actually go up. That's at clinical trial size and demand. So we have no indications that supply will outlive demand.
Okay, thank you for
Thank you.
clarifying. So do we have more questions online from Ryan?
Joanne, how are you defining Becker?
That's a great question. So in the, and it's a hard question. I know that PPMD is grappling with this at this point. It's easy to define diagnosing Duchenne Becker muscular dystrophy. The hard part is distinguishing between the two. For adults with Becker, then the definition is more straightforward, because if they're walking past 18 on steroids or 16 not on steroids, that is essentially the classic definition of Becker. So for adults, that's easy. The harder part is when they're younger than that, and you can't see the future with a crystal ball. So we know that the different kinds of mutation don't tell all of the story. But patients who have an in-frame mutation, so that they have a fully transcribed protein, although it has certain deficiencies in it, are most likely 90% of patients with Becker have that kind of mutation. So for the adolescent boys, that's going to be the criteria, that they have a mutation in the DMD gene with an in-frame mutation.
Okay, I think we have a question over there, and then over there.
I have a question for HOPE-3. The clinical trial says it's ambulatory and non-ambulatory boys, but it also says loss of independent ambulation between 10th and 18th birthday. So how do you qualify the loss of ambulation?
Just repeat your question, please. I'm sorry, it had to do with ambulatory and non-ambulatory and what the definition is.
Correct. How are you defining because the trial says ambulatory boys are eligible. So if they walk less than 1 second for the 10-meter walk, 1 second, less than 1 second per meter on the 10-meter walk, then they're eligible. So if they're able to do it independently, is that still qualified?
No, what we're looking for, it's a non-ambulatory patient population that we're focused on, but we recognize that there's a transition phase that's going on. The transition phase is defined for HOPE-3 as essentially 10 meters in 10 seconds or slower. So we're looking for that 10-meter walk to complete at 10 seconds or slower. That qualifies for the transition phase.
Okay, thank you.
Okay, thank you. We have a question on the other side.
Yeah, I actually have 2 questions. For Astellas, could you clarify what you mean by muscle health in practical application of observation?
I'm sorry, I'm a bit hard of hearing.
No, that's okay. You said that the drug was to protect the muscle and keep it healthy. And what does that mean? Are they staying flexible longer, walking longer, less fatigue? What is it you're measuring?
So it's hypothesized that be able to increase energy production and decrease inflammation, and would slow down the progression of some of these processes that are related to the inflammation and fatigue of the muscle. So, for our efficacy endpoints, we are looking at energy. So it's the cycling, so seeing how many times the patient can cycle at baseline, and then at midway through the study and at the end of the study. So it's really energy that we are measuring for this phase 1b study.
Okay, thank you.
We are also looking at other endpoints as well. You know, I mean, we are looking at other things. We are a part of the imaging DMD study, looking at the fat fraction for the MRS at certain centers are doing that. And then we also have echocardiograms throughout the study and EKGs, but they are not necessarily endpoints, but we will be looking at the data. Okay. And also pulmonary function tests as well.
And then for the CAP 1002, you did mention that they go into open enrollment after for 12 months. And then my question is, what happens when that 12 months ends? Like, do you foresee that you're going to have the drug available, or do they just stop taking it after having it for 2 years?
So the question was for Capricor?
Yeah.
And it had to do with the open label extension, correct?
Exactly, because you said that was for a 12-month period, correct? And so then what happens after that 12 months?
So right now, HOPE-3 is planning for a 12-month open label extension. My belief is past actions are predictive of future actions, and I'll just leave it at that. HOPE-2 actually had a 12-month open label extension also. That's the data that you'll see this afternoon. But after 12 months, that was extended to 24 months. And after 24 months, our plan is now to extend it to 36 months. Okay. So even though I can't tell you that that extension is now on HOPE-3, my belief is personally, and I think the company's belief is that past actions are predictive of future actions. Does that answer your question?
That did entirely, thank you.
Okay, thank you. Are there any more questions online?
Yeah, there's a couple of questions in regards to the fact that a few drugs are going to be before the FDA soon and may be approved, and how they may factor into the ability for those patients once approved. I think it's going to depend on timing of everything, isn't it? But if it's an approved medication, the question is, will they be able to participate if a study is available at the time?
And short answers, please, in view of time.
So for us, we believe that because of the mechanism of action is independent of the potential approval. So again, we anticipate that, you know, as long as the patient has been on the approved product for 6 months or longer, that they could potentially still enroll in our studies.
Approved exon skipping therapies would be automatically included. Others we would consider and amend as appropriate.
And for us, it's going to be essentially an exon skipping dose that's stable. I mean, we want a patient that's coming in where they're at a stable plateau in terms of any impact.
Okay, thank you for that quick clarification. In view of time, I'm going to ask everyone to give a round of applause for the speakers.